All 30 trillion cells in your body have the exact same DNA. But what makes them so different from each other? What makes the cells in your brain different from the cells in, say, your arm or your leg? To explain this, let's go way back to when you were an embryo. Every human being starts out as a little clump of cells. These cells are all what is known as pluripotent. They can differentiate or turn into any of the different cells that make up your body. Cells differentiate from pluripotent to multipotent until they become unipotent. Cell differentiation is a process triggered by gene regulation. You see, all cells have the same basic DNA, but different genes are activated in each of them. This gene regulation is controlled by molecules called transcription factors, which turn genes on and off depending on what type of cell they belong to. So that raises the question, if all our cells come from the same place, how did they know what they should become? How did the cells in your head know that they need to differentiate into the neurons that make up your brain? This is a very complex question that scientists still don't fully understand. It relates to the idea of morphogenesis, or the creation of form. That's the process by which cells know where they should go, what they should become, and how they should interact with each other to form a fully developed body. There are many different factors that we have discovered play a role in morphogenesis. First up, polarity. This relates to the differences in cells based on their orientation. In apical basal polarity, cells facing inwards experience different forces than cells facing outwards. For example, cells on the outer surfaces of your body are exposed to different environments than cells on the inside. This distinction helps guide differentiation. In planar polarity, cells on one part of a flat layer experience different forces than cells at the other end. This is important during the formation of the spinal cord, where cells at the edge of a plane pinch inwards to form a tube. There are many other different types of polarity that are all crucial for the formation of the embryo. Next, morphogenesis is driven by chemicals such as morphogens. These chemicals are distributed unevenly across the embryo. They can affect gene transcription, therefore affecting differentiation. Cells that are exposed to a high concentration may differentiate differently than those exposed to a low concentration. This is what Alan Turing believed created patterns such as zebra stripes. He believed that chemicals diffuse and react unevenly, creating patterns that eventually lead to colorings like these. Cool, right? Finally, mechanical forces also change the way cells develop. For example, when the heart is still in formation, fluid forces created by blood flow influence the way its cavities develop. These forces help the heart and many other organs form the proper cavities that they need to function. Morphogenesis is a really cool process. It's how we can go from one singular cell to all the millions of diverse ones that make up our body today.